live musicians.
Restaurant.
Okay, thank you so much again. I'm really excited to be here. We're here because we're really excited about biology. I wake up every day and I'm just first excited to be alive. It's marvelous. And then second, just thinking about how things work, how different organisms come together, how DNA makes us who we are. And we hope to have a chance to share all of this excitement with you today. We have a series of people that are going to talk. I brought some of my friends from the department and essentially bringing to you a journey through biology, of which we have also written some songs to further enjoy the biology. I want to highlight my bandmates, Daryl Bowling and lead guitar. I'm singing and playing the rhythm guitar. And with that, the first thing I'm going to do is tell you a little bit about the outline of what we're going to do today. The program includes talking about frogs, talking about bacteria, talking about the human microbiome. Yes?
Dr. Catalina, Jen Tarleton is asking if you could please share your screen.
I can. Hopefully it works. Let's see. I don't know what that is.
Click on the continue to hear.
Okay, let's see. Okay, here we go. Frogs, bacteria, the human microbiome. We usually have an intermission. We might take a few minutes, but I don't want to take too long to make sure to make it up to the end of the hour. We're going to talk about pitcher plants and carnivory shortly. Then we're going to talk about epigenetics and getting things a little bit more complicated. And to finish, we'll talk about nitrogen. And I'm really excited about this part because it was mostly inspired by work that we did here at the Marsh. Okay, and with that, we're going to start with frogs. I wanted to ask Dr. Kim and Emery Parker to join us over here. I'm going to share your screen. Here we go. Where's the mute? Oh, I thought we did too, but okay. Okay. All right. We're talking about frogs. Oh. Pretty.
I haven't seen you in a few weeks. I haven't seen you in a herpetology class. And you missed the midterm.
Yeah, I was gonna see, I've been very ill. I was wondering.
Your base is loaded. On top of the temp.
Why did you miss so much class?
Well, I don't want to alarm you, but I think I might have the big C.
Cancer?
No, not cancer.
COVID?
No, not COVID. I think that I might have chytridiomycosis.
What sort of symptoms do you have that makes you think you have chytridiomycosis?
Well, I've had, a cough and some congestion and fever.
I hate to tell you this, but as a larval newt, you don't have lungs to be congested, you only have gills. And those other symptoms aren't typical symptom of the frog itself.
Well, that's a relief. I'm so glad. Thank you for telling.
If you come to class, you would know a lot more about it. And conveniently, we have some slides here. So why don't I go through them and maybe did you have a chance to look over any of your notes from students who were in class? Yeah. Okay. Well, we'll pass it through and see what you might have learned outside of class.
Yeah.
So I and a few others in our department are interested in amphibians and their biology, and there have been a lot of concerns recently because a lot of populations are declining. So multiple reasons that that's true. But one thing that has come to our attention is that there are a number of emerging species that are impacting amphibian populations. And one of them is a fungus. Do you know anything about what kind of fungus causes the disease of chytridiomycosis?
It starts with a B.
That's good. That's a good start. So kindred fungi are a group of fungi that are water moles. So they're small, a lot of them are free living and they eat dead and decaying organic matter. But there are a couple species that actually parasitize the keratinized parts of amphibians. And they have very long names. They both start with a B, the Trachochytrium dendrobatidus and the Trachochytrium salamandrigorians. They're very long, so we usually just abbreviate them, BD and B-Sal. And some have estimated that BD is the more well-studied of the two, that it might have been caused the greatest loss of biodiversity from infectious disease in recorded history. So this is a little bit of an older figure that's showing different places that people have sampled, and then what we usually do is just kind of swab the skin, and then you can use various types of molecular methods to detect the DNA. So where it's in red, that's where it's been detected, and any place we see a dot is where they've sampled for it. Often people just come and they find frogs that look like this that are dead for some reason, and they'll take those back to the lab. They'll also study the skin and try to understand a little bit more about it. So we found rapid declines associated with BD in those. So do you know where we have never found it? What kind? Yeah. So my dad, yeah, we did the ACE exam. Yeah, because there are no amphibians in Antarctica. But every other continent where amphibians occur, they are known to have BD. But not all of them actually get a very serious disease from it. Some of them can carry it without showing any disease symptoms. Some of them can show those and then basically fight off the infection and clear it. So, but the concern is that over 350 species are known to be able to harbor the infection, and about 200 species really do get a serious illness, and that's caused declines, and in some cases, like multiple extinctions of the population. And we've only known about it since the late 90s, so these are really, but we think that some of these unexplained disappearances, especially for salamanders in Central America, likely occurred because of BD. But it is sometimes hard to go back to those historical samples and get that information. So the harm that you think you might be feeling, likely is not BD because it is attacking the keratinized mouth parts. And that wasn't really what you were experiencing. So on tadpoles, actually, there's not much that has keratin. It's kind of restricted to around like the teeth and the beak of the tadpoles. So this is a section the tadpole has taken like this, and the pink area is the stain for the BD fungus that kind of gets actually inside of the cells and then will multiply, and then the spores will kind of break out of this. Just one of the sides. So here you can see those little like kind of openings where the spores will swim out on an infected animal. So for adults, the skin is keratinized, and that's where you typically see this infection. Instead of really nice, smooth skin layers, it gets all all bumpy. Some of it's really thick, some can be thin. It starts sloughing off. And that can affect the water balance in these animals. And in fact, it can impact them so much and change the electrolyte balance that they can essentially have a heart attack because of the change in the concentration of those electrolytes.
I'm not gonna have a heart attack.
I don't know. Wait and see. We'll give you a few weeks.
Suspense.
So, any ideas of where BD came from?
I mean, I'm sure I saw it in my notes. But I think that I'm just so sick, I can't remember.
It's just gone.
It's just gone.
Well, there's still some ambiguity there, but some of the most recent studies sampling different strains of BD from across the world suggest that it actually probably evolved in the Korean Peninsula and probably in like 1920, 1930, and then it was spread across the world as the trade in amphibians became more popular. And the two, when it was originally discovered, it was because of die-offs in both Central America and Australia, basically at the same time, so probably in those like 50 years it kind of made its way around. So there's still people monitoring all of these and still trying to do, basically this is based on DNA sequence analysis, so people are going in and trying to get even more samples to try to rebuild the history of BD. So the one that you might be especially worried about is B cell. And this one is more recent, we know less about it. It was discovered in 2013 in fire salamanders, and it's a little hard to see, but they have a lot of the same symptoms as frogs that are infected with BD, kind of the skin sloughing, and fire salamanders and a few other species in Europe seem to be very susceptible. As far as we know, we don't have it here in North America, but people are constantly monitoring for it. And it seems a little bit more species specific. We don't know a lot, but some of the frogs that are sensitive to BD actually don't seem to be infected with B cell, which in a way is good news. But here are a few of the other species, European species that do actually get chytridiomycosis from B cell.
Wait, but those are new, it's like me.
Yeah, they're in the same family yesterday.
Oh, no.
So keep it away from North America.
This is not good.
And from what we've discovered so far, we think that B cell probably originated in Asia. And this is partly due to the fact that some of these species from Asia actually don't get chytridiomycosis. They don't seem to be infected. So they might have been able to evolve resistance with that for a long period of time. And I don't know if they have a time estimate out. But there's been, again, pet trade to Western Europe, and that's really where we're seeing a lot of the impacts. They're having some studies seeing what it would do to species here, and some of them do seem to be susceptible based on lab trials of ways people are trying to monitor and make sure it does not cross over to our continent. Okay, but you missed a lot of class. So I've got some important questions here, and I feel like if you can answer all these correctly, maybe you'd earn a passing grade. If you can try, these are really important. They frog biology questions.
I think I got it, I really did go over my notes.
Okay, so what is a frog's favorite footwear?
Open-toed sandals.
Okay, now you got 3 more. Think that you can get to answer faster. All right, what is a frog's favorite flower? Rose flower. One.
I think the answer is crocus.
That's what I was thinking. All right, 2 out of 2. Let's see if you can do it. What's those dot croak dash croak dot?
It's gotta be Morse code.
Alright, last one. What kind of music do sophisticated frogs enjoy?
Music. I know, hip hop.
Anybody else have any ideas?
I don't know. Opera. So I guess I'll see you in herpetology next semester.
You get that better.
Thank you so much, that was wonderful, sorry. Cheers out. I have used your folks. Here. All right, so now. Okay. We're gonna sing a song about frogs. The story, so this frog is it's in Spanish. This song is in Spanish. The translation is gonna be on the screen. And I just wanna give you a quick rundown of what the song is about. It's a love story between a male frog and a female frog, where the male frog is immune to the chytridiomycosis, so it cannot get the disease, but the female might not. We'll see. All the frogs then. All right. So now, hold your frog questions for the end. The experts are gonna be around to answer them. And now I wanna transition to Act 2, where we're gonna talk about bacteria.
And
Yeah. I'm gonna be the expert, next time I should have Mark be the expert. But I'm gonna tell you a few things about bacteria, I wanna emphasize I could talk about bacteria all day, every day, for a whole semester, or a lifetime. But I wanna talk about 4 things. One is how small they are, and so how we can think about that scale and that size. How they are everywhere, how they eat everything, and how they move, because I find that fascinating. That's what I spend my time thinking about. So we're let's think about how small they are. And the way I think about this is in terms of scale and how we experience it, right? We have all walked underneath the redwood trees and felt the difference in scale and how small it feels to be next to these giants. Now, you've traveled around California, hopefully, maybe driven to LA or gone to San Diego, right? Imagine your feelings towards California, what it would feel to move from one side, from here, all the way to the southern tip. That's the same relationship of a bacteria that lives on your head and wants to go to your foot. That's the same scale. How you feel about California is how your foot bacteria feel about you. Right? It's kind of crazy. So if we imagine this is my foot and we zoom in, we could see the bacteria and they would be experiencing that same scale. I don't think they think about me very much, but maybe. Who knows? Not pretty cool. And these are just numerically, bacteria can be between 2 to 10 micrometers. I'm 1.5 meters. I don't know about you, but that's a nice size. And California is about 1,200, 1,200 kilometers. And this is just if we put it in the same scale of Earth sizes, how it would look like. So thinking about scale by balancing these numbers, it helps to think about how they experience the world. The second part that I think is key to understand bacteria is to remember that they are everywhere. Taps, everywhere. They are in the mountains, at the top of the mountains, at the bottom of the mountains. They're in the volcano, right, Mark? They're in the water, they're in the plants, they're in all the animals you have ever seen. They're in your house, in your kitchen, they're in your bed. They're in your hands, they're in your mouth, they're in your poop. Everywhere. Every single place you can imagine, you're gonna find bacteria. Places where humans have never been to, the bottom of the ocean, the very tops of the mountains, there's bacteria there. Floating in the air, falling from the rain. We just had a project of students throwing bacteria from the rain. Don't drink it. We can talk about that more. Okay. They're really small, they're everywhere, and they eat almost everything. But I'm not going to talk about what they can eat because they can eat everything, but I want to talk about how they eat and what it turns into, because I think it's a more interesting way to think about bacteria. And so what I want you to do is imagine you have a taco. Tacos are pretty good. You're gonna eat a taco, and when you eat a taco, you put it in your mouth, you chew it, you're happy because you like the taco, and then you go for it and you eat it. Right? And then you feel your belly all happy, and that's it. When bacteria, you use your mouth to eat it. When bacteria eat a taco, or any other resource, the taco is just an example, right? We can talk about anything else, carbon, nitrogen, phosphorus, any material that they eat. Instead of putting it in their mouths, which they don't have, they're going to excrete proteins and enzymes to degrade it outside of their bodies. So most of the time they're doing the digestion outside, which means that they're going to degrade the taco and then slurp the nutrients and the tasty things. The interesting part is that we don't know if they feel happy in their belly because they don't have a belly, but instead when they're happy, they divide, they grow and they divide into 2. So from 1 bacteria, you get 2 bacteria. And if you keep feeding them tacos, what's gonna happen, Emily?
you might end up with 4.
You might end up with 4, they're gonna grow and they're gonna divide into 4, and so on and so forth, more and more and more until you're covered in bacteria, or more covered in bacteria because you're already covered in bacteria. Okay. Last piece, and then I'll sing a song. It's their movement. Bacteria live in a very interesting environment because their relationship to everything is very different. The first part that to me is fascinating is to think that their environment, whether it's air or fluid, is experienced in a very different way. And this is why I want you to think about it. If you're moving through the air, you know, it's easy, right? You go and you walk and it's comfortable, you can move, right? So you can move forward. What if you're in a pool? Say a pool, hip high, it's a little harder, right? And so you like, oh, take a little bit of time to go from one side to the other. Now, what if you're in a pool, but it's full of honey? That's a lot harder, right? So for you to move from one side to the other is gonna require a lot of energy. Fluid and even air for bacteria is a lot more viscous, which is how we describe that force that is going to impede their movement. And so moving is a lot harder. And most of the time, what they're gonna do is use their flagella to propel themselves in these fluids, but the movement is going to be constrained because of those forces. Now, the interesting part is that they're also really, really small, which means that they can float really easily, whether it's on water or air. So they can move. They can't move on purpose really easily, but they can move across continents for free. No need to pay some crappy extra for your bag or like through the air from one side or the other. So I think we're gonna see some bacteria moving really fast in the air. Okay, so hopefully they do for the sound. That's awesome. So hopefully what you got for this little bacteria talk is that they're really, really small, which means that they experience the world in a different way. They are everywhere. They can be found everywhere on Earth. They eat everything, and that allows them to reproduce and duplicate themselves. And they can move really fast through large spans of space, but they can't decide where to go. And now I'm going to sing a song about that. This one's in English. Lift up my hands to and think of all the broken sky. It makes so pretty before you hold your hand. Because I came out I try to think from my head to my feet, and let it try my past, and I think of all the broken sky. When things start moving on, because I had to be my life. Come my fast, then I think all the microbes in time. Anything of your yes, I did. All I think of all my bacteria are claiming some busy life. They really like it. All right. R3. The next expert is me again. And I want to talk about the human microbiome. So the obvious next question is, how do they associate with us? Where do they live? How can you find them? Hopefully you're comfortable with them because you're full of them. And I want to do a quick summary of what we find, where we find it, what they might be doing. The first answer is they're everywhere on the body. They're on your hair, they're on your ears, they're on your skin, they're in your stomach. They're practically everywhere, and they love you. There are 3, it says between 1 and 10 bacterial cells per human cell. So oftentimes I wake up in the morning and I ask myself, am I even human? Or am I just bacteria? I don't know. What's the answer? All of them. The other part is that this is not just a numerical problem. About 1/3 of the metabolites in your blood are actually originated from bacterial activity. So the things that your body is doing are 1/3 driven by what bacteria are doing. And this is all extremely new. All of this research is just now coming out, and you're going to see it in the news coming more and more through the years as we figure out how this affects our health or well-being. Who is in your microbiome? These are some of the most dominant taxol bacteria that you find in general. And they're in colors: actinobacteria, Firmicutes, Proteobacteria, Bacteroidetes. But what I want you to pay attention to is that as you go to different parts of your body, the hair, the skin, the nostrils, the vagina, the stomach, the oral microbiome, the colors of these little pies are different, which means that each of the habitats in your body for microbes are extremely different. Your skin is like the desert. It's salty, it's dry, it's waxy. The stomach is dark, it's acidic, but it's full of food. And so on and so forth. And we can talk a little bit about who specifically is in there. So these are pictures of Propionibacterium acnes. This is a popular one in your skin. Staphylococcus epidermidis in your in your skin and your face, and this one in the rest of your body. It dominates the inside of your elbow. Somehow that's its favorite place in the world. In the oral cavity, you also find Streptococcus, Bacteroides, and Prevotella are in your stomach. And it really depends what your diet consists of. So one of the most important parts is whether you eat meat or you're a vegetarian like me. My gut is probably full of Prevotella. Everybody that eats meats, it has Bacteroides kind of filling that niche in that space. And if you eat dairy, and if you're vegan, and all of these aspects of your diet are going to shift your microbiome a little bit. E. coli is very common in the gut. E. coli is usually your friend. Sometimes it gains a bad rap from the few pathogens that are out there, but most E. coli are your friends. They help us gain vitamins. And then for those of us with vaginas, Lactobacillus species are the dominant taxa in there. It's very acidic and they protect this area of the body from invasions of pathogens. So where do they come from? When we are born from our mothers, we have 0 bacteria. And as we are born, we obtain the bacteria mostly from our mothers. So those of us that were born naturally obtain the first batch of bacteria from going through the birth canal. Those that were born through C-sections obtain their first microbiome from interacting with the skin of the mom and with the hospital or the home or where you are. Breastfeeding is also really important. If you're breastfeeding, you obtain large quantities of bacteria from your mother, and that particularly influences the development of your gut. But what I find most interesting is that from the time you're in the womb inside of your mother, the number of species in your body are going to change. At first, the diversity is going to increase. It's going to reach a maximum when you're an adult, and it's going to decline at the end of your life. Now, the funny thing about the microbiome is that adult phase begins at 3 to 4 years old. That's when your microbiome is an adult. So when Sylvia turned 4, we threw a huge party microbiome adults, because that's when it gets defined as you grow. Now, these are kind of the same as before, little pies that show the differences, and depending on where we're getting the data, but it emphasizes a couple of things about how your life might influence your microbial community. Whether you're born naturally or C-section is the first determinant, whether you're breastfed or formula fed, whether you undergo antibiotic treatments early on is going to have huge determinants on the health of your microbial community. As an adult, obesity, smoking, and how much exercise you perform regularly are keys to a healthy microbiome, and then age. And a lot of the information that is coming out highlights that microbiomes might be associated with health and disease. For example, there's associations with, we talked about diet and antibiotics and smoking, with asthma, with dermatitis, with allergies, with kidney stones, with colon cancer and other cancers, skin, mouth conditions, diabetes, and several different infections. I said allergies. And one of the most interesting ones right now is it's mental health. There's a link between your gut, the microbial communities in there, the functioning of your neural systems in your gut, and your mental health. And if we could tap into this, we could figure out how to manipulate the microbial community in a way that supports stronger mental health. And that would be good. Okay, there's a lot of things that you can do in your life or that you do regularly that are going to change the type of microbiomes that you have. And one of the most interesting, this is a really silly study, but I thought it was fascinating, where they looked at families that had children, that had dogs, that had children and dogs, or neither. And so these are the options. Family with dogs and children, family with dogs, family with children. Which one do you think had the highest number of species of bacteria? Who thinks A? Okay. Who thinks B? Don't cheat, Gavin. Who thinks C? Okay. You said B, Evan? C. You think C? Okay. Okay, C. Okay, so the answer, I'm going to show you a little plot here. This is the diversity. This is time and how they measured it. And so higher bars signify higher diversity of bacteria. And what these results show is that the most important parameter is having dogs. All the families with a dog had higher diversity of microbial communities because the dogs enrich the family's diversity with soil and water bacteria that you wouldn't have otherwise. Unless your child is super wild, which I don't have. But the significant difference was with dogs. So love nugget, you got it. All right, let's sing a microbiome song. There's in your nose and noise, your bowels and all down the slide. There's more than I'm sensation. Now you can live it for you. You can live it for you. If you put it up now, thanks. What's next? Let's hold the questions because we can run out of time. Okay. No information. Let me tell you a little bit about what I do at Cuellar-Gempeler. I'm interested in how microbial organisms associated with animals and plants. And one of the most interesting things that we'll be doing is thinking about carnivory in plants. So how do plants shift the script and eat animals? And how does microbiome in these systems support the carnivore? And these are just different species of pitcher plants, which are plants that produce these modified leaves that attract insects that fall into the traps, drown, and provide nutrients for the plants. All of these plants live in soils that are devoid of nutrients, so they use these animals as a source of primarily nitrogen, but also phosphorus to support their lives. And this is our own California native, Darlingtonia, California. You can find it. You can find it in the zoo, grows right next to the bear cage. There's like three little plants. But there's big populations in Trinity House and in Crescent City and all over the place. So as I said, they have these modified leaves, they attract ants and other insects that fall into, and these insects and animals not only support the plant, but they also support a rich variety of microbial organisms that live inside. So I want to tell you a little bit about it. Bacteria are the main decomposers. They produce those enzymes that break down the ants so everybody can feed on them. But then protozoans and rotifers and mites, they all eat bacteria. So the bacteria are the main, producers in this system. And then there's these mosquito larvae that feed on those protozoans and rotifers. So you have a system with the top predator, like a lion, that is eating little protozoans that is eating bacteria. And you can use this system to ask questions about how the world works, how do forests work, how does the ocean work, and how do organisms relate to one another in other systems. And the beauty of it is that you can go in here and take the sample and manipulate it and play with it and track it through time very easily. The way I think about it is these are small worlds, like ours, but really small. And it allows us to be able to study things that we can't study in the bigger world and try to understand the bigger world through these. So what kind of questions do we ask? We ask, are species moving from leaf to leaf? Does this impact the type of species we find in each leaf or the number of species? How do species come together? How do they coexist? Do they compete with one another? Do they fight against each other? Who's eating who? These are all the things I can't sleep on. I need to know. And one of the most important ones for me is how do they function? How do they get together to break down those insects and feed the plants? And if we can answer this, I think we can answer how any ecosystem works and save the world. So can small worlds teach us about the real world? Am I correct? At least some people have given me money, so they think they're right. That's it. But we have found some evidence. I'm not just telling you things. It's true. We have found things that I'm going to sing in a song. And you have to read my papers, I guess. They're cool. Somebody should read them. Yeah, that's it. Thank you so. Okay. Now I'm tuning, guys. No, I'll show everyone the car. Fashion test. But it's so smart. It's my number and all they are is mosquitoes and horses and new streams. Stay here. They have been black, makes you fight in your life. You can laugh, you can sing, get a place so you don't know it. But if you don't understand, unless you change your time, gotta click song for life. Almost me talking about Thank you. Now let's talk about something very different. When I was in graduate school, I had an assignment, and you're like, oh, okay, I'll get to it. And it was a paper that made me completely change the way that I thought about a lot of things in biology. It was a paper about epigenetics. Who has heard about epigenetics here? Okay, a few people. And it's the idea that you have your DNA with all this information, but it can be changed through your life. And so the paper I read was about rats. And so I thought, oh, that's cool. Is it real? Does it matter? And so I have no idea what the answer is. So I brought an expert, Dr. Amy Sproul, is going to tell us about epigenetics.
Thank you, Catalina. Hi, everybody. It's not nearly as entertaining as the past few presenters, but hopefully you can still learn a little something. So as Catalina just described, epigenetics is what I think to be one of the most exciting and fascinating scientific questions. And I do love amphibians, and I love frogs, and I love bacteria, and I want to live in a pitcher plant. And I think I'm going to try and work that out. But although this is technically a different scientific topic, it's completely related. All of these things are related because of DNA, right? So I know Catalina just asked how many people had heard of epigenetics. How many people have heard of DNA? Okay, so when you think DNA, what's the first thing you think of? Anybody? Diversity. Okay, I get diversity. What else? Reproductivity. I like that. What else?
4 bases.
4 bases. Very nice.
Helix.
I'm sorry, what did you say, Gavin?
The helix. I think it was on the.
And the helix. Okay, so this helix, thank you, Gavin. Excellent. This and all of you, excellent, because everything you just mentioned is absolutely relevant to DNA. So DNA, this very simple helix, which means you can see the helical shape, which looks kind of like a spiral staircase. You can see maybe that there's in the center these doublets of color. And these doublets of color are the 4 bases that one person just referenced. And these 4 bases come together and they're required, they match, right? The colors are always going to be matched. You're always in this diagram, well, model rather, yellow and green versus blue and pink. And these bases in their particular order encode all the information that's required for an organism to grow, divide, and become that organism it needs to be and function appropriately. And if it's done incorrectly, then really bad things happen. One of the big C's that was referenced earlier, cancer, is one of the most famous. And so when we think about reproduction, really, this is the unit that has to reproduce between parent and offspring. The thing that I think is almost even more fascinating than all of those is that the information in these pairs truly prescribe to a multicellular organism how each individual cell knows what it needs to become, where it needs to be, and how it needs to talk to the other. And so when you're first a fertilized egg, right? When that very first fertilization event happens, at that moment in time, all the DNA is completely open. You have full access to all of these base pairs. But as those cells start to multiply, because they have to, we're made of trillions of cells, not just one. As they divide, they get more information and they actually limit the different base pairs that become available to each daughter cell. And so when differentiation is complete, pieces of DNA that are open in your eye, or maybe even in your retina, are completely different than many of the pieces of DNA that are open in your skin, your brain, or your liver. And so what is available has everything to do with what each cell becomes and how it behaves. One of the most important regulators in determining what is and is not available is epigenetics. And so this little carbon molecule, methane group, will come in and bind to a specific base pair. And when it does, it hides it. And it hides it in a few ways. One, you can see, this is not to scale, by the way, but you can see that it's going to hide some of these pieces. And then because it doesn't really like to be around water, multiple methanes are going to then make that DNA bend and hide the 4 bases inside. And those 4 bases comprise different genes. So you hide genes based on the presence of these methyl groups. And there's multiple more types of markers, chemical markers. There's ways that you can package the DNA that require lots of different proteins. But the thing that I think is so amazing is that there have been studies in lots of populations around the world, and it's not just about what your cell needs to know at different stages of its life to do its job. Your DNA will also respond to the environment, and then some of these methyl groups will start coming on in the presence of chemicals, in the presence, or if a person experiences starvation, there's evidence that there are populations that see changes in their methyl groups so that the DNA could respond to that extreme environment. So it's a relatively new field, and I think it is so fascinating. And so I guess that's what I have to share with you about epigenetics. Does anyone have any questions before I say? Oh, sorry. Sorry.
The questions at the end, so we can say,
Question,
we're
yes.
almost done.
All right.
Anything you need? Was mammalian and rats?
Oh, no.
Yeah.
They're everywhere.
Make some amendments to this. Alright. Epigenetics. It's lyrics and songs of doctor. One is but never said like teacher on the front. Which ones are mine? Last one, this was gonna be quick, but we need to talk about that. Before we go, I wanna thank the Friends of Arcata for the invitation to the Arcata March for this wonderful interpretive center and for taking care of nature here. We love it here. We come all the time, and I think it's a treasure of this area. I want to thank you for playing with me because it's so interesting about this. And I want to thank the scientists for joining me and sharing your expertise. You're wonderful. Keep doing what you're doing. It's great. And you for joining us today. It's been really fun. And your bacteria, because they're great. Thank you, bacteria. No, let's applaud you. Thank you. So, what are you made of? Carbon. Well, nitrogen. Excellent. And I would add phosphorus. So most of us, the majority of our bodies, other than bacteria, is made out of nitrogen, carbon, and phosphorus. Out of these ones, nitrogen is particularly interesting because it's usually unavailable in the environment. And so I like to think about how it moves around. First, let's think about where your nitrogen is. Where is most of your nitrogen?
Air.
There's some in your hair.
Air.
Oh, air, there's a lot in the air. Absolutely, there's a lot in the air, in the atmosphere. We can't use it though. We're breathing it right now, and we can't use it. There's some in your hands. Where else? In your body.
DNA.
In DNA, absolutely.
So in the plants.
In the plants. In the plants that we eat. Excellent. In beans. In the bacteria. Absolutely. In your muscles. In the fungus that you eat. That's all the protein that I eat. Beans and fungus. So your muscles, your DNA, your enzymes, all of your enzymes are proteins that require nitrogen in some form or another. So all your body essentially is made out of nitrogen. Where does it come from? When you eat meat, when you eat mushrooms, when you eat beans, all of those products are going to provide the nitrogen that you need to live. Where does it go? In your stomach. It goes into your stomach, absolutely. Thank you. Reese. It gets processed, it gets absorbed, and then what happens with the nitrogen you can't use?
Urine.
Urine. Excellent. It goes out in the form of urea. And all of that urea goes into the pipes and into the water treatment center. And from the water treat, right? Am I pointing in the right direction?
That orange flag.
There you go. And oh, there it is. You can see it. Right then it gets processed. It goes back into the water and all of these ponds in order, and then back into the bin. That was came out of nowhere. So when we release the nitrogen that we're not using, it all goes into water system, whether it's water treatment or rivers and marshes and the ocean. What happens when it goes there? It is our friends, the bacteria, that then use their enzymes to identify it and release it into the air. Most of the nitrogen on Earth is floating in the air. We're breathing it right now. It's like 70% of the air in this room is nitrogen. We just can't use it at all. Who can use it? Bacteria. Thank you. Because most people think that the nitrogen in the air is being fixed by legumes, and they're wrong. It's not the legumes. It's the bacteria in the soil and in the root nodules of these plants that fix the nitrogen from the air and provide it for the plant. What happens is then, once it's fixed, the plant can grow, it can produce little beans, we can eat the beans, the chicken can eat the beans, and then we can eat the beans and the chickens. And then we can be strong. In conclusion, to finalize this, you are full of nitrogen, and bacteria play a critical role in cycling that nitrogen around, without which we would be unable to use nitrogen at all. And by we, I mean all creatures that are alive on the planet. So, what are we going to swim about?
Fix them, always get lost.
Hmm? Never mind. You're gonna hear that it won't. Nothing happens. I was like, what is Stevie so wrong? It's me. Okay, let's go. It's lost in the No, I do that. I made up, broken. I still think I need to when you know. Yeah. Everyone. I think I do that. I swear to make you again. I said right now we provide. Free one you know. Thank you so much for coming.
This was so refreshing.
Thank you.
I have a question about bacteria. Are the main groups of bacteria in my gut, would they recognize as bacteria in the soil?
Ooh, great question. So some of the groups are similar, but if you were to look at the two communities alongside one another, they're really easy to distinguish. So the bacteria that live in your gut are clearly bacteria that live in your gut, even though they're close relatives, and some of them can be even picked up from the soil. But the communities are very different when you look at the whole, it'd be like looking at a rainforest versus grassland. There's plants and some of them are related, but they're super different.
Yeah, okay. Thank you.
Great question.
The frog fungus is the.
Frog.
Is it related to the fungus in the bats, the white
Ooh, good question, expert.
I don't think closely, because the crickets live in water. The bat fungus is dry, cool. But I think they're pretty distant.
I have a question about, you mentioned that some of the food we eat is that, just bacteria in our gut, break it down and it's absorbed into our system because of the bacteria in our gut, the nitrogen and everything else, all the other minerals that we require.
Yeah, I missed the question. Yeah, everything you said is true. A lot of the things that we eat, we can't process them right away. And so there's a lot of enzymes that the microbes are producing themselves that help us process the food or diets. And so some extreme examples is all the animals that are herbivorous are unable to break down lignin and cellulose and all the things that they eat. And so they require their microbiome for their diet completely. They would be completely unable. A lot of the diet, the fiber that we eat, you also need those bacteria. But in addition to the specific things that you're eating, there's a lot of vitamins and cofactors that we can't make ourselves. And so we need the microbes to take our nutrients and make the vitamins that we're missing to have a balanced diet.
Is it true that we fermented foods like sauerkraut and kimchi that you increase the diversity of the microbiome? And then eat certain foods that kill it? And then another question is when you see these advertisements, take this mouthwash, kills 99% of the bacteria, most of the good bacteria. And then it's interesting, like a city like Arcata, it's inviting, has fluoride in its water. And we just allow that without any question, even though there's no good, no harm.
Thank you for bringing both of those topics. I think it's really key that we talk about all of these things to know which one of these ads and which ones of these statements are true and which are not. And so going back to your first question, the fermentation, the microbes in those foods are very particular and have very specialized metabolism. So they do bring new metabolic processes into our gut, and they're usually very beneficial. Also, the byproducts that they're producing are really helpful, particularly if you're already coming from a diet that is not super great, right? So people that just had an antibiotic treatment, for example, or you have a diet that is not very enriched in fibers, getting those fermented foods are going to enrich your microbiome, making it a lot stronger and benefiting your health. Now, in terms of what we're treating, I think it's almost like a shift in the paradigm of how we think about microbes, because for centuries, we thought that we had to kill them all, right? It was like, kill them all, they're gonna kill us, clean it, right? And now we're starting to understand that some of them are beneficial, and we're trying to figure out how to balance those two aspects of defending ourselves against pathogens whilst maintaining the healthy microbiome. And so we're learning to do it, honestly, because
it seems to me in America there's an obsession with cleanliness. So on the counters of the buildings, all these chemicals. And it seems to me if there's some food on the counter or on the floor, you use chemicals you could eat. Because you can't put them out. But everybody gets so many chemicals and the houses stink from it.
Right. It's learning. We're learning. We're in a phase of our human civilization of learning how to live with them while knowing that they're there and figuring out when can they make us sick and when we can coexist.
I have a question for the frog lady. I live in Arcata by a farm at the end of J Street. And they make these tripping sounds. Wait, but if you walk up fast, it automatically stops. And if you walk very, very slowly, like, it doesn't stop. But it gets a certain level, you get to a certain speed, it stops. You have to like creep up to them. I wonder what's happening. The frogs are hearing everything.
Probably the rapid movement, predators move in that way, right? And so they're like, but if you move very slow, then they're like, oh, it's fine.
Let's see over
I
here.
have a question about.
Yes.
Sorry.
I work in a preschool, I volunteer at a preschool, and the kids are always touching each other, hugging each other, kissing each other. I mean, they're 3, 4, and 5-year-olds. Will their skin bacteria become the same over the course of a year or two? Would they have the same environment in their skin?
That's a great question. There's two sides to that question. One is that we know that when we interact with one another, we're exchanging microorganisms. So even if you're not licking each other like 3-year-olds, there is exchange in your partner's skin tends to become more similar to yours over time, just because you're living together. Now, so that would say, yeah, if you're interacting and hugging each other, kissing each other, your microbiomes are going to be affected. They're probably going to be enriched. But the other side of that story is that the skin, as I was saying, it's a habitat that is very inhospitable for, normally dwelling. So the microbe that lives in the mouth and the saliva lives in a moist environment that has a temperature that is pretty stable, is not exposed to UV, versus living in the skin. It's crazy. It's like throwing, a little herb into the desert and seeing. So the overlap is going to be really small. And the final answer is we don't know. Nobody studied that I know 3-year-olds and figure out how their interactions or non-interactions change their microbiome. Really cool questions.
Kids beautiful.
Let's go with you.
Yeah, I have a kindred question. I missed how much of our amphibians are already gone because of it. And are we having any luck selecting or resistant products?
I don't know if any have gone entirely extinct. There's a few species in Central America that they have captive breeding programs for because they think they're pretty much gone. But a lot of them have sort of persisted at low levels, and some of them do seem to be resistant and kind of hanging on. There are some Australian groups that do have captive, like selective breeding programs to choose the resistant ones and are breeding those. I don't know if they've released them back yet, but the same sort of thing where they saw big declines. And I think there might be a captive breeding program for some of the Central American ones in DC. They have a big group there. So it is, but it's a slow thing. And some of them don't like to mate in the lab, which is a big problem. They just will not produce offspring. So then you're kind of stuck with one generation.
So it's early.
Epigenetics question?
How much do viruses rise to epigenetics?
That's an excellent question. I haven't seen a lot of research on viral induction of epigenetics. So the answer is, I don't know, typically the virus will enter the cell and the cell's normal response is to try and digest it into as many pieces as possible and present pieces outwards so that your adaptive immune system can develop the appropriate response and participate. So viruses don't typically interact with DNA proper. But
So the immune system might.
well, so maybe the adaptive immune system, so when the NHC complex presents the pieces of a virus for those T cells, and then the intracellular signaling could do something to the T cell to change the way the DNA is expressed to help. But I have not seen any research on that, and now I'm gonna go look it up. Yeah, there are a couple other epigenetics people and reveal themselves, so choose.
I have a question. A student asked this to me today, and it relates to epigenetics.
Okay.
Outside of the rat paper that you found,
That was.
because at least it's in rats, right?
Yes.
According to the song. Do epigenetics play a role in, this could even be, well, not in bacteria, but in how species compete in ecosystems?
Where is everybody looking? Excellent,
I
so,
don't have an answer.
no,
But I'm not.
I don't have an answer. So bacteria do have epigenetics. They also mark their DNA and they also change the expression of the DNA with epigenetics. But it's really complex to scale it up to the level of the ecosystem, and I haven't looked, but I haven't seen anybody that has had it happen. I have seen regulatory mechanisms through epigenetics to change things like nitrogen, nitrogen fixation or like things like that. So potentially, yes.
Yeah, I think you're talking about histone code, right? So you talk about different ways.
But bacteria.
Well, that would be the type of epigenetics, right? So if that's gonna happen, it would have to be something that. I think there's a possibility.
Do different species that live in an ecosystem, could they outcompete each other, and could that competition come from different epigenetic mechanisms? That's a great question.
Yes, I would guess yes.
Yeah, billions of years, yeah.
But this doesn't require evolution, right? Because you're just turning them on and off. That's the crazy thing, that it can happen in one generation. Mark has an opinion.
No, I think, well, so if it were to happen in nature, I bet the way it would happen is that one organism would silence genes in another organism. Right, they would do something with this.
Such a bacterial thing.
Then the comparison is with imprinted genes. There's male female competition, and so males are trying to turn these genes on, and females are trying to turn them off, or vice versa.
Yeah, I think that's a great, it's a great model, yeah, and again, I mean, the methyl on the DNA is like the most straightforward. I mean, there's tails, the histone code tails, and all the different types of epigenetic marks and how that regulates and poise genes and what's ready to respond. So I think there's likely, and then the other thing, you're talking about behavior in species that have conscious, it's almost impossible to correlate epigenetics with behaviors, but there is a lot of research on intergenerational trauma, and that is embedded in epigenetics. So probably, but this is a wonderful question. Thank you for bringing that. We need a song. We literally need a song. I'm here. I'm giving you this.
Okay, maybe last question, because we have a case of one sleep to go home.
Yeah, I'm remembering a story from Sweden where some scientists discovered a pattern of behavior, I think. And they determined that it was a result of a famine from hundreds of years earlier.
Correct.
A new one just came out, maybe this year, last year, of Syria. So they tracked it in, Syria's been at war, like what, and they clearly, you see the epigenetics through 3 generations that happen now.
Yeah.
Wow.
Okay, well, that's fun. Thank you so much for coming. Thank you guys. They were great. That's really the only new one.
Oh, yeah. I don't want it. Yeah, it's my turn, and I feel like might have a
That's a very fun song.
That's very fun. They go in this funny box.
So these rules runs are
That's why. And don't say it's hard. There are comments in the avoidance class, I was always like, oh, so that's where they get benefited.
Yeah, that's. That's good. I'm scared. I mean, you think like us, I'm protein diet, right? We just have protein diet, get a lot of water. This simulates protein and all that. I love that. I didn't, it's straight down.
Yeah, this is like a bit, yeah, oh, yeah, it's like, I promise you, there's a whole bunch of things that are done. Awesome. Oh, probably, what is it? Why isn't it so? It's sharing on it. Just wait, he's frequent, they would be all up. I mean, even him though, he gets it, like, this is the time he usually start getting it. So he's still. Yeah, people use that, it's very educational.